Good morning, class, and welcome back to our discussion of how sugar is made by plants and algae, and some bacteria use something very similar to this in the form of photosynthesis. Yesterday we were talking about the light-dependent reaction. Remember, photosynthesis is split into two parts: the light-dependent reaction and the dark reaction, or the light-independent reaction. And just a little review of our light-dependent reaction. It occurs in two photosystems, photosystem 2 and photosystem 1 over here. In the thylakoid of the chloroplast, sun's energy is absorbed into these pigments of chlorophyll. That energy is passed along till it reaches a reaction center, and the energy is used to excite and release two electrons. Those high-energy electrons are then sent through a electron transport chain along the thylakoid membrane, generating a proton gradient inside the lumen of the thylakoid, which is used to drive ATP synthase and chemiosmosis. To replace the lost electrons so that new electrons can be energized, water is split, providing two electrons that will then be energized from the sun and sent through the electron transport chain. Once these electrons have used up most of their energy driving chemiosmosis and creating that proton gradient, they are going to be inserted into photosystem 1. And in photosystem 1, similar to photosystem 2, sun's energy comes in, excites two electrons that are released. They go through an electron transport chain, but in this chain what has actually happened is we're oxidizing NADP to NADH, a high energy electron carrier molecule. So instead of running a hydrogen pump, we're actually just using this electron transport chain to run an enzyme that adds this electrons and hydrogens to this NADPH. Those hydrogens come from the splitting of water. As a byproduct of splitting of water, we get oxygen. And then, like I said, these replace the electrons here. These electrons end up here, and now all this NADPH and ATP we have made is going to be used to fix carbon dioxide into that lovely tasting thing called sugar, and all other carbohydrates and really organic molecules that we have in life came from photosynthesis. Even your proteins originally came from the carbohydrates made in photosynthesis, where the plant then adds nitrogens and sulfurs to make different amino acids and proteins for the plant to use. So thank you, photosynthesis, and thank you, plants. So that is the light-independent reaction, or light-dependent. Now let's talk about the light-independent reaction. This reaction is going to occur in the stroma of the chloroplast. So remember, that's kind of like the cytoplasm of a chloroplast. Little review back to cellular respiration. It's the matrix in a mitochondria. But these cytoplasm areas of these organelles or cells is where chemical reactions typically take place. So they're kind of like a witch's cauldron of chemical reactions, I like to think of. Yeah, it is Halloween, so I got witches on my mind. Okay, and in this, what's going to happen? We just talked about carbohydrates are going to be synthesized from atmospheric CO2. So whenever we look at plants, anything like that, most of their mass comes from carbon dioxide in the atmosphere. That should make you wonder what happens as global warming and human increased carbon dioxide enter the atmosphere. What should this do to the plants that are able to survive the extra heat? And the ATP and NADPH are going to be the energy that's used to fix carbon dioxide into sugar. I do want you to write down some key molecules of the carbon cycle, just like we had to remember G3P and pyruvate and acetyl coenzyme A for cellular respiration. These are the three main molecules you're going to have to know the names of that are probably not familiar to you for the Calvin cycle. And the first one is something called ribulose biphosphate. We can call this RUBP. This is going to be a 5-carbon sugar that's going to be used to regenerate the Calvin cycle. it's going to be what CO2 fixes to start this Calvin cycle. So RUBP, really big one. I'm okay if you don't remember that it's ribulose biphosphate, if you can remember RUBP. But ribulose should sound familiar. This is a 5-carbon sugar, ose sugar, ribulose, 5-carbon sugar, very similar to ribose, which you find in DNA. So just a type of sugar that only has 5 carbons instead of 6. But RUBP, definitely need to know that that is the 5-carbon sugar used to regenerate the Calvin cycle. Rubisco is the most abundant enzyme on Earth, so I need you to know that one. And it's going to be the enzyme that actually takes the carbon dioxide and attaches it to the ribulose biphosphate and gives us a 6-carbon sugar that you would think would just be sugar, because that would make a lot of sense, but it's totally not. And it goes through a cycle and then gets split apart again. But Rubisco, very, very important enzyme to know, and it fixes carbon dioxide to RUBP. Interestingly enough, is a very non-efficient enzyme when we get to talk about it later. Sometimes it accidentally doesn't fix the carbon dioxide and it just fixes oxygen instead of CO2. So it's really an inefficient enzyme, but you know, it's efficient enough where its genetics have been passed on throughout most algae and plants, and there's not much difference between them. And even though it's not a very efficient enzyme, it's efficient enough to make life work on Earth. So there are scientists today working very hard to make Rubisco, a version of Rubisco through genetic engineering that is much better and doesn't accidentally make oxygen fixed to it instead of CO2. And if they're successful in that, we're going to have much better plants growing in the future, totally engineered by humans. So I can't wait till next unit when we talk about how we engineer things, because maybe I'm a mad scientist and I like to think about changing the world in my image. That's kind of disturbing. Okay, G3P or GP actually for this class. I don't know why, but certain textbooks changed the name of this. It's really the same thing. Glycerol 3-phosphate, we can call it GP, is a molecule ATP and NADPH act on to form something called a triose phosphate, which can also be called G3P. So we're going to call it GP and triose phosphate. Triose phosphate, very important, is a 3-carbon sugar, or not a sugar, I guess, but it's a 3-carbon molecule that's going to be used to make glucose or regenerated to ribulose biphosphate. So this might be a little confusing, these last two, until we actually go through the Calvin cycle. And that's what I'm going to do with you guys right now, is go through what happens in the Calvin cycle. Remember, this is going to occur in the stroma of the mitochondria. I'll come back to this one. I would rather draw this out than have you guys just look at it all happening at once. So even though I can't draw very well on a computer, I am getting better at this. And we're going to go ahead and draw it. So Calvin cycle, pretty awesome, happening in the stroma of the chloroplast. So maybe I'm going to draw a little chloroplast up here in the ceiling. And we have our chloroplast, and we have these stacks. And inside these stacks is where the proton gradient from the light-dependent reaction would go. The electron transport chain proteins are all along the thylakoid membrane. It's always good to think of where we're at inside a cell. And so when that happens, a bunch of ATP and NADPH is sent out here in the stroma. So this is our stroma. And here, this is where the Calvin cycle is taking course, all throughout the stroma of the chlorophyll. Okay, so the first thing that's going to happen is we're going to have our leaf, maybe, and inside that leaf, that beautiful guard cell we looked at to start the lecture yesterday will open up. CO2 is going to be allowed to come in, and that CO2 is going to be grabbed by Rubisco, and attached to that molecule we just talked about, ribulose biphosphate. So I'm going to start over here with my ribulose biphosphate, which is a 5 carbon sugar. It's actually ring shaped, but we're going to draw it like this because it's a lot easier to understand. And it's a biphosphate, meaning it has 2 phosphates on it. I really don't care if you remember that, but it's good practice to know that they're there. Because remember, these phosphates are going to have energy. They're going to help different processes happen. So this is our RUBP. Okay, so ribulose biphosphate, and it's going to come up and carbon dioxide is going to fix with it through an awesome thing called Rubisco. So awesome enzyme, it's going to grab a CO2, it's going to grab a ribulose biphosphate, and it's going to fix the two together. And we form Rubisco. And these two are going to come together to form an unstable, give myself some more room to work with here. It's going to form a very unstable 6-carbon sugar. So we had 5 here, we've added 1 carbon here, 6 carbons. And this molecule is just very, very unstable, and it needs to split apart very quickly. And I'm going to add my phosphates to it. Hopefully you're drawing this at home as well, much prettier than mine. And so this is just an unstable molecule. So I'm just going to write unstable here. Okay, so we got this unstable 6-carbon molecule. It's quickly going to split into a 3-carbon molecule. Okay. And this molecule is called, for our purposes, GP, or glycerate triphosphate. Triphosphate means that the carbon, that the phosphate is on the 3rd carbon of the molecule. So that's what that means. And this is a glycerate. So it's just the way these carbon molecules form. Definitely not something that we need to know at this point. So that is GP is then formed. So this is unstable. It forms into GP. And then the next step is we have all that energy that has been taken in from the light-dependent reaction, and we have some ATP. And we're actually going to invest 12 ATP at this step. And that ATP is going to work on that GP molecule with the use of a kinase enzyme. Remember, all this is enzyme generated, and we get 12 ADP out of it. I'm not going to draw the phosphates, but they would be coming out of this as well. Okay, along with 12 ATP coming in and working on this, we're also going to have 12 NADHPs coming in. Remember NADHP because P for plant. That's actually not what it is, but it helps me remember. It's a P. And like I said, this is actually a phosphorus, but remember P for plant because the only pathways, metabolic pathways we talk about in any of these biology classes, at least for a long time, NADPH is only seen and used in plants. It is actually in humans, but we won't ever look at one where that's used. Little review for you guys on this. If the NADPH is losing a hydrogen, this molecule is being oxidized and it's actually reducing this GP molecule. And what we get after investing the ATP and the NADPH into this is a molecule called triose phosphate. This is also just going to be a 3-carbon molecule, and it also just has 1 phosphate on the 3rd carbon. The difference between the two is we have put a lot more energy into it. By reducing it, we have added hydrogens, we've added electrons. Remember, those electrons came from the light-dependent reaction. So we've just made this a much stronger molecule, and this is called triose phosphate, or TP. Okay, two things happen with this molecule, and it's going to get a little confusing now, and we'll come back and put numbers in here to explain how this works, and I will also have you watch a video that really kind of explains this all. So we now have made this 3-carbon molecule called triose phosphate, and what's going to happen is 2 of these molecules, you're like, 2, there's only 1 here, and how did we get more than that from here? There's only 2 molecules total. We'll come back to that. But 2 molecules of TP, so 2 of these 3-carbon TP molecules are going to go off and make sugar. Okay, so these are going to be turned into sugar. Okay, glucose to start with, but any carbohydrate can be made out of them. Actually, any fat can actually be made out of them. So 2 of them go off, because if we put 2 together, we have our first glucose sugar. They go off and make sugar, other parts of the cell. But then 10 of these actually are going to go off to regenerate RUBP. Okay, so we're going to have carbon, carbon, another phosphate, and this is TP again. Okay, so some of the TP is going to go regenerate this 5-carbon molecule of, put some arrows in here. It's going to be used to regenerate RUBP, and to do that we also need to add some more energy. So 6 ATP are going to be put into regenerating ribulose biphosphate. Okay, so that is our Calvin cycle. Numbers right now should be really confusing you, because I've only given you a couple numbers here. So I want to go back and go through that one more time and kind of explain how this is happening. So if you remember, our formula for cellular respiration is 6 CO2s plus 6 waters go to 1 glucose, C6H12O6, plus 6 oxygen. So hopefully you are aware that oxygen was made in the light-dependent reaction, water was used in the light-dependent reaction. So in the Calvin cycle, we're going to use 6 CO2. So I'm going to put a 6 in front of the CO2, and so 6 CO2 goes into a total of 12 of this unstable molecule, or no, 6 of this unstable molecule, and then 12 GPs, okay? Because if we split 6 into 2, you get 12 GPs. That makes sense now that we would use 12 ATP, 12 NADHs, and here we would have 12 TPs as well. So we take 2 of those TPs away, which is good because 2 adds up to 6, and we get sugar, and that leaves us with 10 triose phosphates over here. Now this should also be somewhat confusing to you, because if I put 2 3-carbon molecules together, I don't get a 5-carbon molecule of our rubisco biphosphate. And so that's where this is really weird. We draw this as happening as one thing, but really when I say 6 CO2s, this is actually happening at 6 places at the same time going on. So 6 places in the cell, we have the same cycle going on. 6 places in the cell have the same cycle going on. And ultimately, that cycle just makes it easier to regenerate rubisco, RUBP, and makes it so we can continue to make sugar. So I'm going to show a little Lorax cartoon to kind of go through that again. But one last time for your information, how this works is CO2 is fixated to rubisco biphosphate by ribulose biphosphate by Rubisco, forming an unstable 6-carbon molecule that is quickly split into GP. GP is then acted upon by ATP and NADPH to form triose phosphates. 2 triose phosphates go off to make sugar to be used for any carbohydrate, fat, or protein. And then the 10 remaining triose phosphates are used to regenerate ribulose biphosphate using 6 more ATP. That is the Calvin cycle. It is pretty complicated, especially the first time you go through it. It really helps to draw it out like I did here and talk about it as you go through it. Here's pretty much what I said. Again, I would probably write this down in your notes to make sure you have it. I apologize, these are supposed to go one at a time. But outline the synthesis of glucose in the Calvin cycle. So it would be carbon dioxide is fixed to ribulose biphosphate using Rubisco, forming an unstable 6-carbon molecule that breaks down to GP. ATP and NADPH are used to turn GP into TP. TP can leave the cycle to form carbohydrates, while some of the TP is used to regenerate ribulose biphosphate to keep the cycle going. Okay, that is, how do I end my, I want to show you guys a video, but now I'm worried that I can't find my thing. Okay, so we'll come back to the video in just a second. Okay, to help you guys understand how this whole system works together, TED Eds are awesome. Little cartoon. This guy is kind of like the Lorax, showing you the complicatedness of the Calvin cycle. You would never be tested on exactly how this complication works, but I think it's good to see how it works all at once, and I can't draw that. I can barely draw on here. So this should really help you guys out.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1-carbon gas like CO2 into a 6-carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials, a mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons. The initiator, an industrious enzyme named Rubisco that welds 1 carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3-phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just one carbon that gets soldered to one RUBB chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now, a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. Enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources, such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
I love cycles. You guys have a wonderful day. That is it for photosynthesis. We will be giving you a review on cellular respiration and photosynthesis next. Be ready, be good to study, and just remember every time you breathe out, you're sending carbon out into the world. And that carbon may eventually end up in food and back in you. Or even more fun to think about, a dinosaur once farted because of bacteria doing cellular respiration in its gut. And that dinosaur fart released CO2. And that CO2 very well may have ended up in the breakfast burrito you ate this morning or a cup of cereal. And so you very well could be eating dinosaur farts or even their poop or heck, even the dead dinosaur. So I just think that's awesome because that's the way the chemical cycle in the earth. And goodbye.